So, everyone, so you go here, extract todo, extracte, I don't know how you say it. That's it? Yeah. Then, this is what will work. So you can move this to documentos, and then here. So, then, to open R, this is what we'll do the whole time. You click the project. So, click. And then we'll load the packages. It should work. You'll get an error for MSA, that's okay. And you'll get warnings for other packages, also okay. So, to open scripts, the way you do is, I don't know why that's there. Let's move this. Okay, scripts are uno. Then it opens there. Then you can just run. Oops, yeah, run to there. MSA, I'll find some other package we can use, it's okay. And then, so, además avisos is a warning, right? See, that's okay. And it says, MSA doesn't work, also it's okay. So, I just want to make sure that we can do, so I want everyone to write this.
Okay, do you need any more help?
I think we're okay. Yeah, I think it's okay.
Yeah.
It does not work. Okay. Maybe typers. Okay. Okay. Yes, it works. Okay. So once you run that, see if you can write this and see if it works. See if it gives an error. So select empty cars, comma, 1. I'll write it on the board. And it might not work, and then there's a solution. And if it works, put a green sticker. I put some greens on here so that so let's see. Doesn't work? False. Okay. Try to delete MSA. And then run,
MS, ¿no?
Run, and then it's okay to see. So start from the beginning now. So from here. Run.
Pero yo no tengo lo de ahora en la mañana.
Run. Yeah. Okay. Okay, so what we'll do, go to here. So everyone, just delete MSA. Like that. And then you can run. If you get a pop-up that says restart R, that's okay. Restart. Then this should have come ready. So let's see. Yeah, so read
Tenía, yo lo vi que borró MS.
That's weird. This, so that works.
No le digo que repita.
So, did you get to work? So now try to run.
No, ¿eso dónde lo abrí? ¿Recuerdas?
Carpeta R.
Un ratito. Scripts.
Ajá, el primero.
So, what if, okay, if you have it, put the green on there. So, you might need to run this again, the packages library. So run that. Then, see if this works. So, do you have it? Let's see. Let's kind of see you. For some reason, it's. Let's try this. So let's wait. Is not working? Okay, you have to delete the message. So delete. No, it's okay. So just delete that. Yeah. Yeah. Does it work? I see. You have to put the cursor on it. And then run. Okay, so you're good. Oh yeah, I think do your skills. Por favor, if you have it done and this select works, ¿verdad? If you're still working, put this. That way we can make sure we're all good to go.
Yo tampoco.
Put it on your screen, say you're done. Stop. And go. You are. Right, that's good.
Arriba.
I'm good.
Y puedes correr
Good, okay, good.
y ha escrito select
¿Cómo ha escrito
So
select?
you can make a new one or just here. Type that, all goes in and see what works.
Coma 1, ¿no?
Ajá.
Okay, so now working. MSA. Okay, you have to delete. Okay, everybody, make sure in the first line that says packages, delete MSA.
Ah.
So delete MSA. I didn't think that was causing a mistake, but it does. So let's see. So you're good. Okay, again. Okay, let's see.
Delete the
MSA. Yeah. Now run. And then run. And run. Okay, that's good.
Okay.
You too, running. Good. Okay. You started. You're good. Trust me, this will all be worth it. Trust me. It's gonna change your life.
Okay.
Okay.
Okay, you're working. Oh, delete MSA. So delete that and the
Ya,
comma.
ahora como
El
1,
select.
2, ¿no?
Ajá.
Select.
So run to there. Yep.
Ah, es que espérate, has puesto mal select.
Ah, sí.
Might be.
Select. Ah, ok. Ah, ya, chévere. Y ahora sí. ponemos el garbo, entonces, porque estamos
Sí,
bien.
¿no?
Oye, buena, buena técnica esto, ¿no? De los colores.
Sí, ¿no? Todo rojo.
Bien, bien ¿cómo se dice? Interactivo. ¿no?
Y no está
You know? Okay. We can, so we'll make it, so for you, at some point, we'll update our, can you help? Yeah, okay, but you're good, so you can put.
Porque veo marcas de carro.
Gracias.
Sí, no
Gracias,
tiene-
verde.
Claro, pues ese
okay.
MTK,
Uno amarillo.
¿no? Entonces dice MTK, ¿no?
MTK. Es que ellos son como que script ya, por ejemplo el iris también es un script que utilizan bastante cuando te explican el R. Bastante he visto que lo usan. MTK también.
Good job, everybody. Okay, so now that we've figured out, we can use the modern R approach when we come back to coding later in the day. But for now, so Robin will do a lecture on. Oh, this is good, right? Yeah, you're good. You should be very good, right? We're good? Yeah, okay. Robin will do a lecture on sequencing and library preparation, and then we'll go back to the lab and check the PCR, and we'll have, I think maybe some of you do some pipetting as well. Is that good, Robin?
Yeah.
Did you download the lecture?
Well, I just copied it.
You're using the right keyboard?
Oh, of course.
And then we'll need to use, this.
Yeah.
You already do that? Yes, that's good.
Yeah, sorry about that delays and stuff. I think we're back on track. Yeah, so this lecture's in the morning kind of about the background to R coding. This is more about kind of technical, practical aspects, how you actually do sequencing. Main parts of this talk, we're going to talk about kind of background sequencing technology, Sanger sequencing, which was the old method that was used for many years, to what we call NGS or next generation sequencing, which refers to kind of a bunch of more modern platforms. And then I'm going to talk about the one we use, at least in the innovation lab at CVG, the one we're going to teach about in this course, which is Oxford Nanopore Technologies, or ONT for short. I'm going to talk about multiplexing, which we kind of alluded to this morning. This is how you combine lots and lots of samples in one sequencing run. And then I'll go through the library prep protocol in detail that we're hopefully going to do in the lab with you tomorrow, assuming our PCRs work. So sequencing from like the 1970s up until the 2010s, really quite a long time, was something called Sanger sequencing, which probably, how many of you have some experience with this, like looking at these kind of diagrams? Maybe you guys are too young to have done any of these.
Mm-hmm.
Nobody's used to looking at those, okay. Invented by a guy called Fred Sanger. Another fun fact about me, when I worked at Cambridge in England, I worked in the building, not quite the same, but right next door to the building where Fred Sanger worked when he was there. Yeah, he won a Nobel Prize for this. But yeah, it's a method relatively similar to PCR. You have your template DNA, usually amplicons, and you mix them, in this case, with fluorescently labeled versions of the 4 nucleotides. You have a primer, just a single primer to start synthesis, and then a bunch of strands get built where these different labeled nucleotides get built into the strand, and each one is it's what's called a terminator, which actually stops synthesis where it's built in. Building the the fluorescent tag tells you what the base was at the position where it stopped, and then you end up with a series of different lengths of fragments, which you then run through a type of electrophoresis, a laser reads the color, and you end up with these electropherograms in the image. Anybody doing sequencing more than a few years ago is used to looking at hundreds and hundreds of these type of diagrams where each color tells you what the base was in a particular position. And it's still used, this method, it's very reliable, it works well. But cost-wise, it's fixed at about $5 per sequence. And the way this works is like one sequencing reaction just gives you one sequence. So you can't really save money by scaling it up. So when you get to the point where you want to do lots of sequencing, this kind of stops really working. So from about the mid-2000s, we went from really everybody doing sequencing in the same way to a whole bunch of different platforms appearing which are called next-generation sequencing. And all of these really are based on completely different underlying chemistries, which I'm not going to go through in detail because that's kind of outside the scope of this course. But I do encourage you, if you're interested in, to just Google later, these names and get some sort of how they work and what the differences between them are. But what all of these have in common, essentially, is that all of them are very high throughput. They're massively parallel sequencing processes that give you lots of data at a time, not just one sequence. You get millions of reads from an individual run. Some of the platforms are limited to only short reads, like a few hundred base pairs, particularly Illumina and Ion Torrent. They're limited. I mean, there's a few different versions of those machines, but most of them will just give you short reads. Which can be useful for some applications, like eDNA. Sometimes all you need is a short read, and it can give you tons of data very cheaply. But for others, we want longer reads. For example, that's too short to give you the full-length barcode fragment. This is just an interesting thing that I throw in. There's a thing called Moore's law, mainly applied in like computer technology, which is about how costs in technology tend to decrease through time. It was sort of created to do with computer processing power and computer memory, that it sort of decreases in a linear log fashion. And sequencing actually violates this rule. You can see when Sanger sequencing was the norm, the cost per raw megabase of sequence was pretty much following a line matching the trend. But then right in the mid-2000s, the technologies completely changed and the cost savings, it became suddenly far, far cheaper for every megabase of DNA that you want to sequence. So costs have really come down dramatically for doing sequencing in the last few years. As I say, this is the platform we're really going to be talking about in this course, Oxford Nanopore. It's part of what's called the third generation of sequencing technologies. It can give you very long reads, like just about unlimited really. There's no sort of inbuilt limit to how long of a read it can get. It can give you thousands, even I think they have done millions of gene pairs. People have done entire chromosomes with this technology. It reads native DNA. It doesn't require any sort of amplification or synthesis. So genomic DNA can be sequenced directly by this method. You don't actually have to do PCR. It can read RNA as well. It can also read base modifications like epigenetics, methylation and deaminases. Not really relevant to barcoding as well, but that's an important application in things like cancer research. This is used for that. So their first sequencer launched in 2015. I just showed you what it looked like this morning. It's portable, it's about cell phone size. The real advantage from the point of view of biodiversity is that it's a lot cheaper than a lot of other sequencing platforms. It's about $1,000 for each sequencing device, the MinION, which is an 800th the cost of the PacBio Sequel 2. That costs about $800,000. PacBio Sequel 2, by the way, is the main sequencer that most of CDG does most of its sequencing on, actually, which is a really high throughput, really fast machine. But for kind of innovation for smaller projects, Nanopore is long way. The library prep, compared to others, is quite straightforward, it's quite fast. It takes about 2 hours to go from your PCR product to your sequencer. I'm hoping we can get that whole thing done tomorrow afternoon and running on the sequencer. The most significant disadvantage that it has is it does have a slightly higher error rate compared to other NGS platforms, but we have ways of addressing that bioinformatically. It's still more than sort of 99% accurate, but that means that every so many thousand bases you get will potentially be wrong. So there's things you need to do to get around that. And this is sort of a diagram of how sequencing actually works. You have these pores which are part of the device. A DNA strand gets wound through it and converted to a series of electrical signals, which the system can convert it to what the length of the bases were. I'll show you that in a bit more detail. The DNA fragment needs to have something called a motor protein attached to it, which is what the library prep is essentially, is attaching those motor proteins to the DNA so that they can hook onto the pore and get drawn through. The pores exist on these flow cells. The flow cell is a sort of cartridge that is what actually gets plugged into the sequencer. And it has, well, 100 or so in the case of the small flow cells and thousands in the case of the larger ones. Individual pores, which is constantly drawing the DNA through and generating these electrical signals which are recorded and captured by a computer. And it can just connect to an ordinary computer like a laptop or something, connect to iPads and things like that. The flow cells themselves come in a range of different types. The smallest one is called a flongle. I have a few examples here. Yeah, maybe we can. I don't know. We could just pass these around the lab. I think these are used ones, so it doesn't matter. So that one is the flongle, which is the smallest one. Costs about $100 US, and it gives you about for the barcode size somewhere between 500,000 and 1 million reads. So even the smallest one is giving you a lot of data. The next size up is the mid-ion flow cell. That's the kind of standard one for this size of device, the medium size device. Costs about $900 per flow cell.
You can pass.
Yeah, $1,000, or you can have some cost savings by buying at the same time, but that gives you like 10 million to 15 million reads. The larger one, which we don't have the sequencer here, I don't think Dorothy has one, but the Prometheon is the sort of larger size of the device. The sequencer itself is a bit bigger. It's about the same size as this bag here. But the flow cells actually themselves, they're a different shape, and they actually cost about the same amount of money as the mid-ion flow cells, about $1,000. And these give you actually 10 times again as many reads as that one. You can get 100 million to 150 million reads. So really huge amounts of data, probably more than you need for barcoding applications typically. But these are what we tend to use for metabar coding, when we're resequencing big, complicated mixes where you want to get lots and lots of sequences, lots of reads. And these are often used for genome sequencing as well, whole genome sequencing. Yeah, how does the library prep actually work? Just kind of to go through this. So you have your template DNA. And just to be clear, this is assuming we're sequencing a PCR product here, as we're generating at the moment. It's going to have all of the primers and the tags in them already, which I haven't shown on this diagram, but just bear in mind that those are in there. The first thing is you do something called end prep. That is an enzyme, and it just comes as part of the sequencing kit that you get when you buy this. And what that does is put a 3' A, an adenine base, on the end of every fragment, and what that's doing is making them compatible with ligation. Ligation is simply sticking one piece of DNA to another piece. It's done with an enzyme called DNA ligase. And these sequencing adapters are how you put the motor protein on. That's this yellow blob that I put on here. Again, this is just something that you buy with the kit. These have a 5' overhanging T, and that is the sort of compatible sticky ends that allow one bit of DNA to get stuck onto another end, which I'm using. And so you end up with adapter ligation. This is what we call the final library prep DNA. It's got a motor protein on each end. Now, only one strand is what actually gets sequenced, but it could be either strand randomly. It's just whichever motor protein happens to find a pore when it goes through the sequencer. And once you've done this, that's your final library. You do this in a series of different tubes, different enzymatic reactions, and that is what gets loaded onto the sequencer, which you're going to see tomorrow.
Robin,
Yes.
in the red sequence is the tag, yeah?
The tag, yes. Well, no, it's not the tag. It's the sequencing adapter.
Okay.
That is what connects the motor protein. There is a method of doing this to get tags, like you mean for multiplexing
Okay.
tags. There is a way of doing this with that as well, but that's kind of the next part of the talk. this point is this is just how you get the motor proteins onto any DNA, regardless of what it is. Yeah, multiplexing, we've kind of been alluding to this a lot, I haven't really been in detail what it is. So, as I say, any NGS method will give you millions of reads per reaction. So the way to get sort of economic use out of that is to load lots and lots of samples onto the same reactions. You get lots of reads at the same time. But in order to do that, you need a way of separating them afterwards, because all you get out of the sequencer is just millions and millions of reads, and the sequencer doesn't know what they are. The way you do this is by adding what we call unique molecular identifiers, or UMIs, to each individual sample, or your specimen. And what these are synthetic sequences that we've designed ourselves that will map to an original sample. A funny thing to bear in mind is that for most of the world, in next generation sequencing, most of the community calls these things DNA barcodes, because they're barcodes to tell things apart. Obviously, in our specific community, DNA barcodes mean a different thing. They're tags for biodiversity. But just bear in mind that if you're reading about next generation sequencing in a lot of other contexts, medical contexts and so on, when they talk about DNA barcodes, they're talking about this stuff. They're talking about the things you put onto sequences to identify them. So yeah, we are calling them UMIs, but that is kind of our CBG or barcoding community specific terminology for these things. And what a UMI is, they are. It just means that in a plate, let's say this is an original PCR plate, you get a bit of DNA that we've designed ourselves gets added onto every single DNA molecule that we generate, so that we can then just mix them all together, pool them, run them on a sequencer, and then every read that comes out, it will have that tag as part of it, and therefore we can tell, or a computer analyzing this will be able to tell, okay, this read maps back to excuse me, this read maps back to A1, A2, A3, and so on, all across the plate. How do you do that? Well, there's various different ways of doing that, but broadly falling into two categories. One of them is ligation based, and there is a kit from ONT, it's called the native barcoding kit that allows you to do this. And that, what you're doing there is just simply using ligation, very similar, kind of as you were mentioning, Manuel, just now. Essentially the same method that you use to put the motor proteins on. You also use DNA ligase at an earlier step to put on these unique tags. And then you do once you've done that, you can do a second ligation reaction on everything to put the motor proteins on, because that's what you have to do to sequence them. And the nice thing about that is it's extremely flexible. You really can multiplex absolutely anything you want, any kind of DNA. It doesn't need any special primers or adapters or anything like that. The disadvantage is that library prep for that is more complicated, because you have to do that end prep step and ligation step for every individual thing that you want to multiplex. So this is a nice approach if you've got just, say, I don't know, 10 different things that you want to multiplex. Once you get up to an entire 96 well plate, this is getting a bit old. You don't want to be doing that many separate ligation reactions and read cleanups on everything. The other approach, and this is the one that we're doing in our experiment today, the one we spend most of our time doing in our lab really, is PCR-based indexing, which is where the UMIs are tagged onto the primer sequences. So as I was mentioning this morning, anything that you put onto a primer automatically gets built in to every amplicon that gets made with this primer. So we can use this. Primers, of course, need to be designed in advance. That means you need to order lots and lots of different primers. You need to have as many different primers as there are things you want to multiplex. So it's quite a big upfront cost. But once you've done that, library prep is really very easy, because all of the indexing you need to do, it's already done, just by doing the PCR. This is the idea here. So here we have a forward primer. Let's say that's the CO1 forward primer. That's the same for everything you want to do, because that's the bit that actually has to be complementary to CO1. But then every primer has this UMI built onto it on the 5' end, and they can be all different things. These are things we design ourselves. Same for the reverse. And so every amplicon ends up looking like this, with the primer, and then on the outside you've got your UMIs, which tells you where the sample came from. And there's a few different ways you can do this. The UMIs can be symmetrical, which essentially means you just have the same information on the forwards and the reverse primers. In some ways, this is just a simple system. All you need here is 96 unique sequences. The unis we use, I think they're about 15 base pairs long, so you can easily get enough to have 96 unique combinations of letters in that. And the reverse, you have the same information on the reverse end, although it appears in the reverse complement direction, obviously, if you're just reading one strand. And this has the advantage that bioinformatically you only need to see one end in order to assign any given read to its well. But the disadvantage is you can't multiplex more than one plate in this web well. You need to design as many unis as things you can multiplex. That gets hard very much. What you can do is use native barcoding on top of this to multiplex more than one plate together. We've done that as well. But this is the system we more often use because of how kind of simple it becomes when you have a lot of things to do. Asymmetrical unis. This is where the uni on the forward and the reverse primer are different from each other. Forward, our system at least, it can go either way around, but in our case, the forward primer is exactly as I showed you before, it is 96 unique uni sequences. But then on the reverse end, the reverse primer it's one sequence for every plate. So everything within a given plate gets the same reverse primer, but we can have a series of different reverse primers, and we can use that to multiplex a whole lot of different plates together. Bioinformatically, that means you need to see both ends, you need to see both complete ends of a read to see both unis. But if you get that, the reverse primer gives you the mapping to a plate, and the forward primer gives you the mapping to a well. So forward is always, it's the same 96 repeated across multiple plates, but then the reverse primer gives you mapping to a particular plate. That's kind of here showing you how it works. So our system, you can do more than 10 plates actually, but in the sort of system we see sending around the world, because most labs are not doing huge numbers of things. This lets you multiplex 960 unique combinations of primers with 96 forward and 10 reverse, which if you do one negative control per plate, lets you do about 950 specimens, which is a good amount for multiplexing in one sequencing run, like on a flow notebook. So as I say, each of these plates has a different reverse primer, and each well has a different forward primer, so that's how that works. Yeah, this just kind of reiterating here. So the amplicons end up looking like this. This is your actual data from your organism. The forward primer, the reverse primer is here. Bioinformatically, the forward primer tells you the read direction, because of course a read could come in either orientation. And then the forward uni tells you the well, and the reverse uni tells you the plate, and these are the motor proteins. Yeah, so our method overview, it's kind of small, I don't know if you can read this. But it's showing we can make this a relatively fast process. Alkali lysis, the protocol actually usually takes about 10 minutes. I've got 5 minutes written here. Amplification of the DNA with our pre-made PCR plates, as I was showing you, takes about 2 hours, or just a little bit over. Adding the necessary adapters, because we don't need to do any further indexing after that. It's already done by the PCR. All we need to do is to put the motor proteins on. That takes an hour and a half to 2 hours. You load it on the MinION sequencer. So for these kind of numbers of samples, we use a flow cell, the smallest one. One takes 24 hours. And then the bioinformatics. You do quality filtering, demultiplexing, which means identifying the unis, identifying things to the samples that they should be. Adapting and removing primers. Yeah, there's a lot of bioinformatics steps, which I'm not going to go into too much in this course. Actually, this lecture, they're going to be part of other lectures. So just mention what we've done, and that takes about a couple of hours as well. So we're talking about in a couple of days' work, you're going from samples to results. Yeah, the library prep, just to look at that in a bit more detail. Firstly, you pool all of your PCR products, all of your different reactions into one tube, which the first time I did it just seemed the most wrong possible thing you could do to me. Against every molecular biology instinct that I have to take multiple reactions and mix them all together. But no, you do it and you can separate them by the data at the end. Yeah, you do a cleanup with magnetic beads, which I'll go into in a bit more detail in the next slide. You quantify how much DNA you have, because you're supposed to load an approximately constant amount on the sequencer. You do the ends prep, as I mentioned, that's making things compatible with ligation. You have to do another magnetic bead cleanup after that. And then here's your optional step where you do ligation of native barcodes if you want to do that. Another bead purification after that. You get really used to doing bead purifications if you do this protocol. And then whichever one you've done, whether it's direct or with native barcodes, you do adapter ligation, as I was mentioning, that's putting the motor proteins on. One more bead purification, and then quantify how much of your final library you have, and then that gets loaded on the sequencer. Yeah, we do have this published as a protocol. This is on protocols.io, and it actually gives details for actually this one tells you how to do up to 24 96 well plates. It's got details of primers for all of that. But yeah, this has got the entire library protocol in it. So if you kind of want to look up the detailed protocol for that, it's there. I don't think anything's changed in that compared to what I'm going to tell you. Okay, yeah, step one, simply pooling all of your plates, however many plates it is, get pooled into one tube. Strange as that seems to me. You mix with these magnetic beads. Ampure XP beads is one brand name, or Spry is the sort of generic name for these. It's like solid phase reversible normalization. And what these are, each of these are little particles. Each one has a kind of iron core at the center that's magnetic, and it's surrounded by carboxyl groups. And what those do is they weakly bind any DNA that they're mixed with in the right binding environment. You need something like polyethylene glycol also mixed in there, which is what's called a crowding agent. And that binds with water molecules, and it stops DNA from being soluble in water, and therefore forces it to kind of stick, precipitate around these magnetic beads. Once you've done that, you hold a magnet up to the tube, where you put it against a magnetic rack, and then all of your magnetic particles with the DNA attached get pulled out. And then you can just take out all of this liquid, the supernatant, with a pipette. You do a couple of washes, usually with ethanol, which you do because DNA is insoluble in ethanol, so ethanol will not wash out the DNA, but it will wash out a lot of salt and other stuff, impurities that you want to get rid of. And each time you do that, you just pull down again with the magnet, take off all the liquid that's there, the DNA will stay safe as long as it's on the magnet. And then the final step is called elution, where you add either a buffer or just water, which solubilizes the DNA. And as soon as the DNA is in a sort of soluble environment, it will just fall right off the beads. Or with a bit of mixing, it will come off the beads, and it's in solution again. Pull them down to the magnets to get the beads separated, and then you can just take that off, take it to a separate tube. And this is it's a very good method for it. It gives you very clean DNA. It gets rid of nearly everything else, and you can also use it to concentrate DNA way down, because you can elute in whatever volume of water you want, and then your DNA is just dissolved in that. Yeah, end prep, as I mentioned, pretty simple. You just take a fixed amount. We usually go with about 100 nanograms of DNA to an end prep reaction. You add this enzyme and its buffer, which comes from New England Biolabs typically. You do this step in the thermocycler, which is 20 degrees for 5 minutes and 65 degrees for 5 minutes, which just inactivates the enzyme once you're done with that. Another mag bead cleanup. I won't go through that again, but it gets rid of all the end prep buffer and all that stuff. Next step is you add the DNA ligase enzyme with its buffer and the sequencing adapters. Those are the things that have the motor proteins on them. That reaction, very simple. It's just room temperature, about 25 degrees for 10 minutes. I sometimes do it in a thermocycler anyway on incubate, just because you can be extremely precise about what temperature they're seeing then. And 25 degrees is the optimal temperature for that enzyme. You do a final mag bead cleanup of your library DNA. That's your final library now. You quantify it. There's a process of priming the flow cell. There's a sort of preparation solution that you load onto the flow cell before running it, which I'll show you as we get to that point. And you load your final library. So for a flow cell, the small one, the recommended amount is 10 femtomoles, which for of course, moles to grams, you convert with, it depends on the length of the fragment. So for 650 base pairs of CO1, that's about 5 nanograms. So you don't need very much. You typically get enough from one PCR pool would be enough to do potentially dozens of sequencing reactions if you want to. 5 nanograms is not a lot. For the larger flow cell, it's about twice as much, 10 nanograms. And from there, it's setting up on the computer. This is software called Minnow, which is software that Oxford Nanopore have produced themselves, and we'll see. I think we're gonna do the sequencer on the computer that we brought with us, but this is just something you can download for free on whatever computer you need the sequencer on. And then you just leave it to run for about 24 hours, and the next step is bioinformatics, which I guess is going to be the subject of future talks. That's my last slide. Yeah. Any questions about any of that? Was it too fast? Or? Okay, I think we can get ready. Is there anything else you want to talk about from here, Ken, or should we head down to the lab now?
Yeah, I think the first time I saw these protocols, it was all a lot of steps, but you'll do it, and you'll watch Robin do it, and then it'll all start to make a lot of sense to you. So, yeah, I think we can go.
A question.
Question.
Yes.
Do you use a library ligation or another kit for library?
The kit is Oxford Nanopore Ligation Sequencing Kit. Is the name of the kit. Yeah, they make a few different kits for different kinds of sequencing, but yeah, that's the one we use. It's probably the most standard one.
Rapid?
It's just no, rapid is a different thing. That is based on tagmentation or something like that. That's not the one we're using. No, the basic kit is just called the ligation sequencing kit, and it's got the motor proteins and buffers and things that you need with that. Now, it doesn't actually come with DNA ligase or end prep, which seems a bit strange because it has ligation in the name, but no, you have to buy the DNA ligase and end prep enzyme, usually from New England Biolabs. That's where you buy them from. But yeah, Oxford Nanopore's protocols give you the lists of everything you need.
Why we have to do two quantifications and we quantificate with metabar or by fluorescence?
Sorry?
The quantification, the flow
Quantification. Yeah, the
Y2.
white. It's only really the last quantification that's important because you have to be quite specific about how much of your library you load on the sequencer. So that's kind of the important one. We generally do a quantification after cleaning up the initial PCR pool as well, and that's kind of just a confirmation that we have some DNA there, and to give us some rough guidelines of how much we want to put into the library prep. But the library prep is actually fairly flexible. Like, so honestly, so long as you have kind of a minimum of about 50, and it doesn't really matter how much DNA you use, it will probably work. So yeah, the first quantification you could probably skip if you wanted to, but the last one immediately before you load on the sequencer you do need to do because it's quite precise about the amount that should be loaded on.
Anything else?
How many samples can we sequence you need?
Okay, so on a flow roll, the most we've done is, I think, the 24 plates, so that's 2,000 and something, like 2,200 samples that would come to. So that's kind of pushing it to the edge because that, let's say you get 500,000 reads, that's, I can't do that in my head, it gives you a matter of hundreds of reads per individual specimen, but then you do lose a lot because of filtering out low quality reads and so on. But yeah, that generally gives you enough data that you multiplies samples. And the larger flow cell, you can go way beyond that. We have a publication, I should have put the paper up actually, but we showed where we used the combination of native barcoding with PCR-based barcoding to do 100,000 specimens on a amino acid run.
Yeah, so you can say maybe 1,000 barcodes,
Probably 1,000
on
is
here?
the number.
then
Flow cell, yeah.
100,000
Yeah.
on here, and then 1 million.
Yeah, we actually have done
On
up to
here.
1 million with like doing data barcoding twice basically to
Yeah.
create more complications. Yeah, it's quite.
But you never use the
Most
flongle.
people
Oh, sorry.
don't do that.
So you would not use the flongle for metabarcoding. You'd use one of these
Yeah.
or eDNA.
Yeah, metabarcoding is a bit different because each sample potentially contains hundreds or more, different taxa. So you need lots of those. Yeah.
Okay.
You all okay? Okay. Well, then that's a lot.
So
do I have a text or anything?
yeah, it just is. So now we will go down to the laboratory again for maybe 2 hours, 1.5, 2 hours.
Yeah.
And then we'll run a gel to see that the PCR that you did earlier worked. And then Robin has a couple other things planned to practice pipetting and things like this. I think, Robin, if we can.
Yeah, I think there's more DNA samples there, although it sounded like there's not a full place.
We can check it. Yeah.
Were you working on that, Leah, or I can't remember who was working on that? Yeah, since up there. Do you know how many samples it is in total?
27?
27. Okay, so we can do like another partial.
And then after the lab, around I think 5 p.m., 5 in the afternoon, we'll come back here for one more computer lesson. That everything will work, I hope, this time. And then that's it for today. So we'll go to the lab now and start in 10 minutes. Okay.